Good afternoon, friends. I'm Saul. Today I'm with a new video which contains a part of the chapter 4. So, under this part 2, bioenergetics, which includes cellular respiration, that includes glycolysis, tricarboxylic acid cycle, and the electron transport chain, are discussed. So, let us proceed. This is chapter 4, part 2. In chapter 4, part 1, we try to discuss about enzymes. We discussed about some properties of enzymes. We discussed about some class of enzymes. We discussed about mechanisms of actions of enzymes, as well as we try to discuss some factors that affect enzyme catalytic actions. So in this part of chapter 4, I will try to discuss in detail about bioenergetics as well as biosynthetics. This bioenergetics means production of energy in living organisms. Mainly it is about respiration. How to harvest or produce energy in the form of ATP from food which we eat will be seen under bioenergetics. In biosynthesis, mainly it focuses on photosynthesis, how carbohydrates such as glucose and the starch will be produced in photosynthesis process in living, especially living organisms, especially in green plants, will be seen in detail. So this is cellular respiration. Most living organisms obtain energy by breaking down of organic molecules. These organic molecules are mainly macromolecules. They are polymers. So these polymers must be broken down into their respective monomeric units. This is in catabolism. Good example of catabolism is cellular respiration. Therefore, in cellular respiration, larger molecules will be broken down and the respective monomeric unit will be produced, and after energy in the form of ATP will be obtained. Cellular respiration can be divided into three phases. To obtain energy from food that we eat, to get energy in the form of ATP, cellular respiration passes through three phases. The first phase is breaking down of large complex biomolecules. Large complex biomolecules like polysaccharides, proteins, and lipids into their respective building blocks or monomers. These large and complex biomolecules will be broken down into their respective building blocks or monomers by a process known as hydrolysis. Hydrolysis means by adding water, these larger molecules will be broken down into their respective monomers or building blocks. In this stage, there is no production of energy. So this is the first phase of respiration. In phase 2, building blocks that are obtained from larger and complex biomolecules are oxidized to a common intermediate products such as pyruvate molecules by a process known as glycolysis. So in phase 2, glycolysis will be dealt in detail. In this phase, every small net energy in the form of ATP can be produced. From one glucose molecule, two ATP molecules can be produced in glycolysis or in this phase 2 of cellular respiration. In phase 3, this consists of citric acid cycle, which is followed by electron transport chain or oxidative phosphorylation. In citric acid cycle, there is oxidation of acetyl coenzyme A to carbon dioxide in the formation of NADH and FADH2. This NADH is reduced to form of nicotinamide dinucleotide, and FADH2 is reduced to form of flavinamide dinucleotide. This reduced form of molecules are electron carrier molecules. We'll see them in detail later. In electron transport chain or in oxidative phosphorylation, energy in the form of ATP is formed. That means most energy in the form of ATP is produced in this phase, in electron transport chain or in oxidative phosphorylation phase of cellular respiration. The ultimate goal of cellular respiration is to harvest energy from carbon compounds such as glucose, and they used that energy to make energy in the form of ATP. So this ATP is used to provide immediate energy for cells to perform a number of activities, including chemical reactions and any other different activities that cells carried out. So this ATP energy is very important cell for their activities of various ways. Cellular respiration occurs in two main parts. Those three phases of cellular respiration are included into two main parts, glycolysis as well as aerobic respiration. Glycolysis is anaerobic respiration or anaerobic process which doesn't require oxygen. In anaerobic condition, there is no need for oxygen. However, aerobic respiration includes kreb cycle or citric acid cycle, or you can say tricarboxylic acidic cycle, and electron transport chain. This kreb cycle or citric acid cycle or tricarboxylic acid cycle and the electron transport chain all together are aerobic respiration, so they require oxygen. Glycolysis occurs in the cytoplasm of the cell, whereas the aerobic respiration part of cellular respiration is occurred in the mitochondria of the cell. These are main difference between aerobic and anaerobic respirations. So next we'll see detail about these two parts of cellular respiration, the glycolysis as well as aerobic respiration. This glycolysis is anaerobic respiration because it doesn't require oxygen. Glucose is a key metabolite in metabolism. Glucose can be broken down or oxidized into energy in the form of ATP. Or this glucose can be converted into a polymer known as glycogen. This is ATP. But this glucose molecule is obtained from starch. And also it can be obtained from amino acids. By the way, glycogen can be converted back to glucose. So this glucose molecule plays a key role in metabolism. Glucose is either converted into polymer known as glycogen, or it can be broken down into energy products such as ATP. However, glycogen is a polysaccharide stored form of glucose in animals, and It is abundant in liver and the straight muscles. For example, skeletal muscles and the cardiac muscles contain large amount of glycogen. When glycogen is depleted, more glucose is synthesized from starch, and it is also formed from amino acids by a process known as gluconeogenesis. So glucose can be obtained from starch. Glucose can be obtained from amino acids by a process known as gluconeogenesis, but glucose can be oxidized into energy products such as ATP, and this glucose can be converted into glycogen, and even glycogen can be broken down back to glucose. Therefore, this glucose plays a major role in metabolism. This glycolysis takes place in the cytoplasm of both prokaryotes as well as eukaryotes. It is the first step of degradation of glucose molecules, because glucose is degraded or broken down to produce energy. So the first step in degradation of glucose is glycolysis. This glycolysis doesn't require oxygen because it is anaerobic respiration. It results in 2 molecules of pyruvates, because these pyruvates are 3 carbon compounds, whereas glucose is 6 carbon compound. Therefore, 1 glucose molecule is split into 2 pyruvate molecules in glycolysis. This glycolysis consumes 2 ATP molecules, but produce 4 ATP molecules. So the net is 2 ATP molecules. 4 minus 2 is 2. Therefore, it consumes 2, but it produces 4. Therefore, the net product is 2 ATP molecules from 1 glucose molecules in glycolysis process. Glycolysis involves 10 enzymatic reactions. Here, glucose, which is 6 carbon sugar, it gets 1 phosphate group. This 1 phosphate group is obtained from ATP, adenosine triphosphate, and the adenosine triphosphate is changed into adenosine diphosphate, because 1 phosphate is attached to glucose at its carbon 6. Therefore, glucose is converted into glucose 6 phosphate in the first reaction. This first reaction is catalyzed by an enzyme called hexokinase. In second reaction, glucose 6 phosphate is converted into fructose 6 phosphate. As you know, glucose is aldehyde, whereas fructose is ketose. Therefore, they have the same chemical formula, however, structurally they are different. So there is isomerization reaction was carried out. By the way, this reaction is reversible reaction. Glucose 6 phosphate can be converted into fructose 6 phosphate, and the fructose 6 phosphate can be converted into glucose 6 phosphate. These reactions can be catalyzed by the same enzyme. Phosphoglucose isomerase is an enzyme that catalyzes these reactions. Third reaction, there is addition or consumption of another 1 ATP molecule. Therefore, fructose 1,6 phosphate is converted into fructose 1,6 bisphosphate, meaning 2 phosphate molecules are attached at carbon number 1 and the carbon number 6 of fructose molecule. Then this reaction 3 is catalyzed by phosphofructokinase 1. Phosphofructokinase 1 catalyzes this chemical reaction. This fructose 1,6-phosphate, or you can say fructose 1,6-bisphosphate, is split into two molecules. In this way, it is split into glyceraldehyde 3-phosphate molecules. Glyceraldehyde 3-phosphate molecule, by the way, fructose is 6-carbon compound, whereas glyceraldehyde 3-phosphate is 3-carbon compound. This glyceraldehyde 3-phosphate has one phosphate molecule on its carbon 3. On the other hand, this fructose 1,6-bisphosphate is converted or split into dihydroxyacetone phosphate. Dihydroxyacetone phosphate is another 3-carbon compound, which is this glyceraldehyde 3-phosphate is aldose, whereas dihydroxyacetone is ketone. Therefore, this fructose 1,6-bisphosphate is split into 3 molecules. These are glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. However, this reaction is catalyzed by an enzyme known as aldolase. But dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate. Therefore, this fifth reaction is catalyzed by triose phosphate isomerase, because isomerization reaction is carried out. This glyceraldehyde 3-phosphate is aldose, whereas dihydroxyacetone phosphate is ketose. Therefore, ketones are converted into aldose. So this is isomerization reaction. So triose phosphate isomerase catalyzes chemical reaction. So now we have two molecules of glyceraldehyde 3-phosphate. Two molecules of glyceraldehyde 3-phosphate. Therefore, whenever we talk one of these glyceraldehyde 3-phosphate, we are talking about another one. In the sixth step, glyceraldehyde 3-phosphate lose its hydrogen. Therefore, here two NAD plus. This oxidized form of nicotinamide dinucleotide molecule come and take two hydrogen molecules. So what is produced? 1,3-bisphosphoglycerate is or are formed. This reaction is catalyzed by a process an enzyme known as glyceraldehyde 3-phosphate dehydrogenase, because hydrogen is removed. That's why the reaction is dehydrogenation. In step 7, this 1,3-bisphosphoglycerate obtain or give one ATP, one phosphate group for ADP. Here, ADP, adenosine diphosphate, is here. So whenever one phosphate molecule is attached to adenosine diphosphate, adenosine triphosphate is produced. So each of these 1,3-bisphosphoglycerates are losing one phosphate molecule. This reaction is catalyzed by phosphoglycerate kinase. Step 8, 3-phosphoglycerate is converted into 2-phosphoglycerates. Then 2-phosphoglycerate is converted into phosphoinopyruvate by losing water molecule. This is enolase is an enzyme that catalyzes this reaction. Finally, phosphoinopyruvate is losing one ATP molecule, one phosphate molecule. Therefore, ATP is formed, and finally, two pyruvate molecules are formed, and this reaction is catalyzed by pyruvate kinase. We'll see each of these reactions in detail in the next slides. Okay. To add more detail about glycolysis, so this is step 1. Step 1, glucose is converted into glucose 6-phosphate. Glucose is converted into glucose 6-phosphate, meaning one phosphate group is attached to glucose at its carbon 6. This reaction consumes one ATP molecule. This phosphate molecule or phosphate group is obtained from this ATP adenosine triphosphate molecule. So the glucose is changed into glucose 6-phosphate. It acts to keep the glucose concentration low. Whenever glucose is converted to glucose 6-phosphate, then the concentration of glucose is becoming lower and low, promoting a continuous transport of glucose into the cell through cell membrane transporters. These membrane transporters are proteins that transport materials like glucose through cell membrane. Therefore, they are suitable with glucose 6-phosphate, not with glucose alone. Therefore, glucose to be transported through plasma membrane. These transporters are very important. So these transporters need glucose to be ionic form. Therefore, to change glucose into ionic form, it obtains this phosphate molecule. These blocks glucose from leak out due to the charged nature of glucose 6-phosphate. If glucose is allowed to pass through the cell membrane, it leaks out of the membrane. However, this glucose 6-phosphate is charged molecule, therefore it is easily transported through transporter proteins without leaking out of the cell membrane. This reaction is catalyzed by an enzyme known as hexokinase. Hexokinase, or it is isoenzyme of glucokinase. So in this way, glucose is converted into glucose 6-phosphate in the first step of glycolysis. Step 2 of glycolysis is the conversion of glucose 6-phosphate to fructose 6-phosphate. These two molecules have the same chemical formula, but they differ in structure. So the change in structure is known as isomerization. So this glucose 6-phosphate is an aldose, whereas fructose 6-phosphate is a ketose. Therefore, aldose can be converted to ketose by isomerization reaction. This reaction is freely reversible in normal condition. It means glucose 6-phosphate can be converted into fructose 6-phosphate. Normal condition, it is a reversible reaction. However, because of this fructose 6-phosphate is constantly consumed by the next step of glycolysis, more the forward reaction is further. Therefore, more of glucose 6-phosphate is converted to fructose 6-phosphate. This reaction is catalyzed by enzyme known as phosphohexoisomerase. This is an enzyme isomerized isomerase enzyme type. Specifically, it is phosphohexoisomerase that catalyzes the conversion of glucose 6-phosphate. Even the reverse reaction can be catalyzed by the same enzyme. The third step of glycolysis is phosphorylation of fructose 6-phosphate to fructose 1,6-bisphosphate. In this step, there is another energy. The form of ATP is expenditure, because the second phosphorylation is necessary to allow the formation of two charged groups that must needed in the subsequent steps of glycolysis, ensuring the prevention of free diffusion of substrates out of the cell. That means this glucose, fructose 6-phosphate is phosphorylated into fructose 1,6-bisphosphate. Bisphosphate meaning there are two phosphate molecules are attached to the fructose molecule at its carbon number 1 and carbon number 6, and this helps us this molecule in the next steps. This molecule will be split into two molecules. So each of these molecules will contain one phosphate group. This helps them not to be diffused fully out of the cell. Therefore, in this step, this fructose 6-phosphate is phosphorylated, or it gains one additional phosphate molecule to change it into fructose 1,6-bisphosphate. This reaction is catalyzed by an enzyme known as phosphofructokinase 1, especially for animal cells. But it can be also catalyzed by an enzyme known as pyrophosphate dependent fructokinase. This enzyme is mostly found in animals. In some bacteria, archaea, and protists can have this enzyme, the second enzyme. However, animal cells will use the first one, phosphofructokinase enzyme, can catalyze in animal cells fructose 6-phosphate to fructose 1,6-bisphosphate. Well, this is a fourth step of glycolysis. This fourth step has two parts, so you can consider as step 4A. It is the cleavage of fructose 1,6-bisphosphate into dihydroxyacetophosphate as well as to glyceraldehyde 3-phosphate. These two molecules, dihydroxyacetophosphate and glyceraldehyde 3-phosphates, are they are 3 carbon compounds. They are trios. So in this step, this molecule will be split into these two molecules. Fructose 1,6-bisphosphate is split by aldolase into two trios 3 carbon sugars. So dihydroxyacetophosphate, which is ketose, and glyceraldehyde 3-phosphate, which is aldose. So from one fructose 6-bisphosphate, one ketose dihydroxyacetophosphate, and one aldose, which is glyceraldehyde 3-phosphates, are formed. There are two classes of aldolase. This reaction is catalyzed by an enzyme known as aldolase, but there are two classes of aldolase. First class, class 1 aldolase, is present in animals and plant cells, whereas class 2 aldolase is found in fungi as well as bacteria. Step 4B is a isomerization of dihydroxyacetophosphate to second molecule of glyceraldehyde 3-phosphate. These two molecules have the same chemical formula, but they are structurally different. So in this step, the structure of dihydroxyacetophosphate is rearranged to the glyceraldehyde 3-phosphate. So this is done by an enzyme known as triose phosphate isomerase. Triose because it works on 3 carbon molecules that contains phosphate group, and the other reaction is known as isomerization. So triose phosphate isomerase is an enzyme that catalyzes the rearrangement of dihydroxyacetophosphate to the glyceraldehyde 3-phosphate. Triose phosphate isomerase rapidly interconvert dihydroxyacetophosphate to another glyceraldehyde 3-phosphate that proceed further into glycolysis, because the subsequent or the next steps of glycolysis require not the ketose group, rather they require the allose molecule. That's why this dihydroxyacetophosphate, which is ketose molecule, is converted into glyceraldehyde 3-phosphate. Of course, this reaction is a reversible reaction, however, the forward reaction is more favored. The fifth step of glycolysis is dehydrogenation and phosphorylation of glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. This glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate, and two reactions are carried out: dehydrogenation as well as phosphorylation. The aldehyde group of the triose sugar, the aldehyde group of this molecule is or are oxidized because they lose their hydrogen atom, and the inorganic molecule is, inorganic phosphate is added to this carbon. So this group is oxidized by losing its hydrogen, and it gains inorganic phosphate. As a result, 1,3-bisphosphoglycerate is produced. Containing 3 carbon atoms, and it contains phosphate molecules on carbon number 1 and carbon number 3. The hydrogen is used to reduce 2 molecules of NAD+. These NAD+, nicotinamide dinucleotide, are reduced. This is oxidized form, so whenever it gains hydrogen, it is reduced into NADH. By the way, this NAD+ needs 1 hydrogen atom and 1 electron to be reduced into NADH. That's why this hydrogen plus or proton is formed, because this proton loses its 1 electron, and the electron is received by this NADH. So, in general, in this step, glyceraldehyde 3-phosphate is dehydrogenated. That means it loses its hydrogen from its aldehyde group. Similarly, phosphate molecules, inorganic phosphate group is added to the same carbon. As a result, 1,3-bisphosphoglycerate as well as NADH molecule. By the way, this molecule is very important molecule in the next steps of respiration. Therefore, these two molecules are produced in this step. 1,3-bisphosphoglycerate as well as NADH are produced. This reaction is catalyzed by glyceraldehyde 3-phosphate dehydrogenase. By the way, mostly the name of enzymes and the name of reactions that are catalyzed by such enzymes are interrelated. The reaction is known as dehydrogenation. So the enzyme that catalyzes reaction is dehydrogenase. Specifically, these enzymes are glyceraldehyde 3-phosphate dehydrogenase, because dehydrogenation is occurred on glycerol 3-phosphate molecules. This sixth step of glycolysis is the transfer of phosphate group from 1,3-bisphosphoglycerate to adenosine diphosphate or ADP. And this reaction is catalyzed by an enzyme known as phosphoglycerate kinase. And this enzyme is magnesium ion as a cofactor. At this step of glycolysis, 2 new molecules of ATP are synthesized, because there are 2 1,3-bisphosphoglycerate molecules are there. Therefore, from each of these molecules, 1 ATP molecule is formed. Therefore, at this step, 2 ATP molecules are produced, and as a result, 3-phosphoglycerate is produced. This reaction is a reversible reaction. However, the forward reaction is always encouraged, because this 3-phosphoglycerate is continuously used by subsequent or the next steps of glycolysis. Step 7 is isomerization of 3-phosphoglycerate to 2-phosphoglycerate. These 2 molecules are isomers. They are chemical. They have the same chemical formula, but structurally they are different. Therefore, this 3-phosphoglycerate is isomerized to 2-phosphoglycerate by shifting phosphate group from carbon 3 to carbon. From carbon 3 and to carbon 2. Immediately, this molecule 2-phosphoglycerate is formed. This reaction is catalyzed by an enzyme known as phosphoglycerate mutase. And the reaction is a reversible reaction, and both forward and backward reactions are catalyzed by the same enzyme, but always the forward reaction is encouraged, because this 2-phosphoglycerate is consumed or used by the subsequent steps of glycolysis. In step 8, there is dehydration of 2-phosphoglycerate into phosphoenopyruvate. Whenever water molecule is lost from 2-phosphoenopyruvate, immediately from 2-phosphoglycerate, immediately phosphoenopyruvate is formed. So 2-phosphoglycerate is converted into phosphoenopyruvate by an enzyme known as enolase. This enolase is an enzyme that requires magnesium ion as a cofactor. And finally, phosphoenopyruvate is formed. From 2-phosphoenopyruvate, 1 water molecule is removed. So whenever 2-phosphoenopyruvate are used, 2 water molecules are removed. So here in this step, 2 water molecules are removed. The ninth step of glycolysis is the formation of pyruvate molecule from phosphoenopyruvate. This phosphoenopyruvate is converted into pyruvate by losing the phosphate molecule. It is dephosphorylated. Therefore, this reaction is catalyzed by an enzyme known as pyruvate kinase. 1 molecule of ATP is generated during this reaction. From 1 phosphoenopyruvate, there is 1 pyruvate and 1 ATP molecules are formed. But from 1 glucose molecule, there are 2 phosphoenopyruvates are produced. Therefore, from 2 phosphoenopyruvate, there are 2 molecules of ATP as well as 2 pyruvate molecules are formed. This reaction is substrate level phosphorylation. By the way, substrate level phosphorylation takes place in two steps of glycolysis. So in substrate level phosphorylation, there is a formation of energy in the form of ATP. So in general, from one glucose molecule, 4 ATP molecules are formed. ATP. From one glucose molecule, 4 ATP molecules are formed, whereas 2 are consumed. So in general, net of 2 ATP molecules are produced from one glucose molecule. Still, there are 2 pyruvate molecules are produced or obtained from one glucose molecule, pyruvate. These molecules are used in the next step of respiration, especially in citric acid cycle or in kreb cycle. These molecules are joined cycle and energy will be produced. So from one glucose molecule, still 2 pyruvate molecules are produced. Similarly, 2 NADH. These NADH, energy carrier molecules. NADH. 2 NADH molecules are generated from one glucose molecule. These NADH molecules will be taken to the citric acid cycle and further production of energy in the form of ATP. Therefore, in general, 2 ATP molecules are produced from one glucose molecule, and 2 are used, and there are 2 ATP molecules are found as a net. 2 pyruvate molecules are still produced from one glucose molecule, and 2 NADH molecules are still produced from one glucose molecule. The second part of cellular respiration are tricarboxylic acid cycle and the electron transport chain system. These two together are aerobic respiration because they require oxygen, or they need oxygen, and they occur in mitochondria. Compare with glycolysis. Glycolysis is anaerobic respiration, which doesn't require oxygen, and it occurs in cytoplasm of the cell. But here, these two cellular respiration parts occur as they require oxygen, so they are aerobic respiration types, and they are occurred in mitochondria. Each pyruvate, which are results of glycolysis, enters into tricarboxylic acid cycle for complete oxidation. So this part of respiration needs these pyruvate molecules. Then pyruvate molecules will be taken into this cycle for complete oxidation. The tricarboxylic acid cycle, another name for tricarboxylic acid cycle is kreb cycle, or you can say citric acid cycle. It is considered as a central pathway of aerobic metabolism because it serves as two purposes. It can be used as a bioenergetics as well as can be used as a biosynthesis. So because of these reasons, it can be considered as a central pathway for aerobic metabolism. So what is bioenergetics? This cycle carries out complete degradation of acetyl coenzyme A to carbon dioxide, GTP, which is energy containing molecule equivalent to ATP, and reducing powers. These are NADH and FADH. By the way, these molecules contains or carries energy. Therefore, in this cycle, energy containing molecules are produced. So that's why it can serve as a bioenergetic mechanism. In other words, this biosynthesis is the cycle, the citric acid cycle or tricarboxylic acid cycle can serve as biosynthesis because many precursors for the formation of other molecules can be made in this cycle. These precursors can be used for the formation of other molecules, for example, alpha-ketoglutarate and oxaloacetates are used for the synthesis of many amino acids like glutamic acid, aspartic acid. Second, succinyl-CoA, which is a part of the citric acid cycle and used for the formation of pyrophorine ring. This molecule can be used for the formation of cytochromes, chlorophyll, as well as hemoglobin. Therefore, pyrophorine ring is formed from succinyl-CoA as a precursor. Oxaloacetate can also be used, converted to phosphoenolpyruvate, which is a precursor for glucose in gluconeogenesis pathway. Glucose can be formed from other molecules like amino acids by a process known as gluconeogenesis. But this oxaloacetate or oxaloacetic acid can be used as a precursor for the formation of glucose molecules in a process known as gluconeogenesis. Acetyl coenzyme is the starting material for fatty acid biosynthesis. This acetyl coenzyme A is used in citric acid cycle as a molecule, and it is also used as a precursor, as a starting material for the formation of different fatty acids. So, in general, this citric acid cycle or carboxylic acid cycle or kreb cycle can be used as a biosynthetic mechanism, and it can be used as a bioenergetic mechanism. Bioenergetic mechanism as well as biosynthetic mechanism. In bioenergetic mechanism, energy-containing molecules such as GTP, NADH, and FADH2 are produced. In biosynthetic mechanism, different molecules that can be used as a precursor for the formation of other molecules can be produced. Steps in Krebs cycle. Prior to Krebs cycle starts this pyruvate. First reacts with coenzyme A and it forms two carbon compound or intermediate called acetyl coenzyme A. Acetyl coenzyme A and at the same time this carbon dioxide as well as NADH are released. Therefore, in this reaction in which pyruvate is converted into acetyl coenzyme A, there are NADH as well as carbon dioxide are released. By the way, this reaction is sometimes considered as part of Krebs cycle. However, it is a bridge that links the glycolysis with Krebs cycle or citric acid cycle. It is a bridge. Therefore, once this acetyl coenzyme is formed, immediately it is taken into the mitochondrial matrix of the cell. To elaborate Krebs cycle, the acetyl coenzyme A reacts with the oxaloacetate. When these two molecules react to each other, citrate is formed, and this citrate loses water molecules. So immediately isocitrate is formed, and this isocitrate loses carbon dioxide, decarboxylated. So when it loses carbon dioxide molecule and when this NADH, which is oxidizing agent, takes hydrogen atoms from this isocitrate, then alpha ketoglutarate is formed. So this alpha ketoglutarate again loses carbon dioxide, or it is decarboxylated. Still, it is also reduced again by this reducing agent NADH plus. So this NAD plus, so and NAD is changed into NADH. So the alpha ketoglutarate is converted into succinyl-CoA. Then on this succinyl-CoA, many reactions are occurred. For example, succinyl-CoA loses the acetyl coenzyme A. One. Second, it gains phosphate molecule. It gains phosphate molecule. So this phosphate molecule joins with GDP. This GDP, guanine diphosphate. When it receives phosphate molecule, immediately it is converted into GTP, which is equivalent with ATP in its energy content. So after this all carried out, as a result, succinate is formed. This succinate molecule is formed. On the succinate, this fat, another oxidizing agent, takes the hydrogen atoms from succinate. So succinate is converted into fumarate. This fumarate also receives water molecule. When it gains this water molecule, then fumarate is converted into malate. Then this malate also oxidized by this NAD plus oxidizing agent, and it results in oxaloacetate. So oxaloacetate is formed again. So in this way, this Krebs cycle is going on, and the cycle repeats itself. However, there are many things are produced here. For example, from one Krebs cycle, 6 carbon dioxide are produced. 6 carbon dioxide, because here from one cycle of this Krebs cycle, there are 2 carbon dioxide are produced. On the bridge, 1 carbon dioxide was produced. So immediately from one pyruvate, there are 3 carbon dioxide are produced. However, one glucose molecule has 2 pyruvates. Therefore, a total of 6 carbon dioxide are produced in Krebs cycle. Second, 2 molecules of GTP are formed. 2 molecules of GTP. So this GTP is already equivalent with ATP in its energy content, and there are 8 NADH are formed. 8 NADH molecules are formed from one glucose molecule in Krebs cycle, because here 1, 2, 3 NADH are formed here, and in the bridge, when pyruvate is converted into acetyl coenzyme, 1 NADH are formed. So a total of 8 NADH molecules are formed from one glucose molecule, and there are 2 molecules of FADH2 are formed. FADH2. By the way, this NADH and the FADH2 are very important molecules in the next step, because they are electron carrier molecules, and they are very essential in the electron transport chain system. FADH2. So these are main points. This NADH and FADH to move to the to play a significant role in the next stage of the respiration or aerobic respiration. Electron transport chain, which is the final step of aerobic respiration, and it is a part at which most of ATP molecules are produced. For instance, from one glucose molecule, there are around 36 to 38 ATP molecules are expected to be produced. However, 2 from glycolysis plus 2 from kreb cycle in the form of GTP. Then after here 4 ATPs are produced out of the electron transport chain. So a total of around 32 to 36 or 34, I think. 34. Therefore, from 32 to 34, ATP molecules are expected to be produced in the electron transport chain. So most of ATP molecules are produced in the electron transport chain of this aerobic respiration. Electrons and hydrogen ions from NADH and FADH2 are used to convert this ADP to ATP. This hydrogen, NADH and FADH2 carry hydrogen atom, and this hydrogen atom is dissociated into hydrogen ion, which is proton plus the electron, because hydrogen has one electron and one proton. So immediately when this hydrogen atom is dissociated into hydrogen ion and electron, this helps this ADP to be converted into ATP, because there is addition of phosphate group. Then when phosphate and ADP molecules are react to each other, ATP molecule is formed. Electrons from NADH and FADH2 move along mitochondrial membrane from one protein to the other. These proteins in mitochondrial membrane that help us electron to move through are known as cytochromes. So through cytochromes, electrons are moved from one to the other and reach to the final destination. This NADH and FADH2 that are obtained from kreb cycle as well as from glycolysis, especially the NADH is also obtained from glycolysis. Therefore, these molecules bring hydrogen atom to the electron transport chain, but this hydrogen atom is dissociated into electron as well as hydrogen ion. So hydrogen ion is pumped out of the mitochondrial matrix, but electrons are transported through this cytochromes from one cytochrome to another. But at the end, electrons are accepted, or finally electrons are accepted by oxygen. However, these hydrogen ions are pumped back into the mitochondrial matrix through ATP synthesis. This ATP synthesis is an enzyme that catalyzes the formation of ATP from ADP and phosphate ion or phosphate molecule. So when these two molecules are react to each other, ATP is formed. Then these hydrogen ions are joined back with electrons and hydrogen atom is formed. Then when hydrogen atom is joined with oxygen, then water molecule is produced. So in this manner, during respiration, water molecule is produced. The hydrogen ions pumped into the mitochondrial matrix across the inner mitochondrial matrix membrane. This oxygen is the final electron acceptor in electron transport system in cellular respiration. The protons and the electrons are transferred to the oxygen to make water. The electron transport, a total of around 32 or 34 ATP molecules are produced from a single glucose molecule. But this is only for electron transport chain. In glycolysis, there are 2 ATP molecules are formed. In kreb cycle, in the form of GTP, 2 molecules are of GTP or equivalent to ATP molecules are produced. But ATP, electron transport chain alone produce around 32 or 34 ATP molecules. Thank you for watching, and I will come back soon with the third part of this chapter. See you again. Bye bye.